Okay, let's explore about lipids in this video. Okay, lipids are naturally occurring hydrophobic compounds which is found in plant and animal tissues. Okay, so the term hydrophobic, that means fear water. Okay, it is water fearing. So in other words, they are not soluble in water. Okay, lipids are not soluble in water, but then it is soluble in other organic solvents such as alcohol, ether, and also chloroform. So like carbohydrates, let's look at the element. They are made up of carbon, hydrogen, and also oxygen elements, just like carbohydrates, but with a much higher ratio of hydrogen atoms to oxygen atoms, which makes them hydrophobic, having these characteristics. Okay, so let's look at the types of lipids. They are fats, waxes, phospholipids, and also steroids. Okay, so fats and oils, they are triglycerides. So we look at the first category. Okay, so fats and oils are the triglycerides. So what is triglycerides? It's actually a type of ester formed from condensation of one glycerol molecule with three molecules of fatty acids. So these triglycerides can be hydrolyzed again. So that means it is broken down, can be broken down, the terms lyse, okay, in the presence of water into its subunit just now we have mentioned, and that is the fatty acids. Okay, the three molecules of fatty acids and also one molecule of glycerol, and that is by means of hydrolysis. So glycerols are a type of 3-carbon alcohol that contain the 3 hydroxyl groups. Okay, that's glycerol. Okay, so this is one molecule of glycerol with 3 molecules of fatty acid. And you look at the arrow, which means this building up triglyceride, the type of ester, okay, it's being formed and 3 water molecules are being removed in this process of condensation, building up. But then when the arrow goes the other way, so to say that the triglycerides is now going to be hydrolyzed in the presence of water, it is being broken down. Okay, you will get the subunit again, which is one molecule of glycerol with 3 molecules of fatty acids. Okay, so this is test for lipids. Okay, the presence of fats can be tested with the ethanol emulsion test. Ethanol is added, so the formation of a white emulsion, okay, this is the emulsified, this is the emulsion that is being formed, okay, shows the positive results of a presence of lipids. So the right, the one on the left, which means this tube, boiling tube, forms an emulsion where the oil is dispersed within the water. So within the water, you see dispersions of these droplets of oil. But on the right side, okay, you can see these 2 distinctive layers, which oil being less dense than water is seen to be floating on top of water, this layer. Okay, and it's a test to show the presence of fats. Okay, so 2 types of fatty acids. Okay, go back to the previous equation. So now we discuss about fatty acids particularly. Okay, so the subunits of fats, bear in your mind there's glycerol and fatty acids, and now under fatty acids we will be looking into 2 types, namely the saturated fatty acids and the unsaturated fatty acids. So let's look at the similarities. Since they are triglycerides, They are fats, so they contain these elements C, H, O. Okay, since they are triglycerides, they both contain glycerol fatty acids, and they are non-polar molecules. Okay, they have these hydrophobic properties. So these photographs are showing foods, okay, that are rich in lipids. Okay, comparison in this manner, the differences, okay, in the aspect of differences, you can see in terms of the single bond between the carbon. Okay, so look at the C. So between the carbon themselves, single bonds. Fatty acids only have single bonds between the carbon, but for unsaturated fats, by the name itself unsaturated, you get to see fatty acids having at least one double bond between the carbon. Since they are unsaturated, meaning this double bond is still able to receive one or more additional hydrogen atoms, okay, because they are not saturated, unsaturated. Okay, unlike these saturated fats with no other places available, no empty place available, so they do not form chemical bonds with additional hydrogen atoms because there's no more place. In other words, we say all the bonds between the carbon atoms are saturated. And because of that, they exist in solid form at room temperature, unlike the unsaturated fats or the oils, they exist in liquid at the room temperature. So here you are, these olive and also fish oil, they are examples of unsaturated fats. Okay, but for saturated fats, you expect to see them as solid at room temperature, and for example, butter and animal fats. Now, the other four category, now we're discussing, we are here, fats, the types of lipids, fats we have covered up. So subsequently, we are going to look at waxes, phospholipids, and also steroids. So let's look at wax. Wax contains one molecule of alcohol that combines with another molecule of fatty acid. Okay, by the name wax, which is like the one in the candle, okay, it is waterproof. For phospholipid, okay, you have heard of this term before, that's the major component of plasma membrane. Okay, when you did chapter 3 previously, you come across the structure of the plasma membrane, and you know the major component, okay, is depicted by those remember the head and also the two tail, and that's the phospholipid. Okay, so phospholipids are made up of one molecule of glycerol that combines with two molecules of fatty acid, and don't forget here phosphor is already telling you that there's one phosphate group there. So one glycerol, two fatty acids plus another phosphate that forms phospholipid. Okay, for steroids, they are lipids that do not contain any fatty acids. So examples of steroids, cholesterol, and also these are steroid hormones. For example, testosterone, estrogen, and also progesterone. Okay, so let's look at the importance of lipids in the cell. It serves as a reserve energy for animals. Okay, reserve energy if there is any shortage of carbohydrates. Okay, let's say no longer store of glycogen, or we say glycogen store has been it is finished. No more storage of carbs in the form of glycogen in animals. So our body is going to make use these fats as our reserve energy. So in addition, fats also function as a liner, okay, to protect the internal organs, also as heat insulator for animals. So you can compare this thick skin and thin skin. Okay, so thick, the subcutaneous fats, you can see that has got what do you call the wider region, okay, as compared to the thin skin having lesser subcutaneous fat. Okay, so it is deposited underneath the skin. Okay, cutaneous here refers to the skin. Sub means below, beneath the skin. So that's where the fats are being stored as adipose tissue. Okay, if the storage of glycogen storage in the liver is full, that is how this excess glucose is being stored. Okay, so continue. Wax. Okay, the second category, we have wax. Okay, they are an important component in cuticles of the leaf. Remember, this is the cross-section of the leaf. In chapter 2, we've come across this picture. So the topmost, that is actually the cuticle layer, and that is a waxy layer. Okay, you see, wax is waterproof. So from the picture here, you can see if water droplets are to drop onto the surface of the leaf. So what are you going to see? Okay, so it's very important to serve as regulates the moisture, okay, that the leaf is exposed to. And without that role alone, what will happen is that the leaf, maybe it will dry out. Okay, besides as a moisture barrier, it also acts as a protector against viruses, against pathogen. Okay, so we say the main function of cuticle is to prevent water loss, okay, and to prevent contamination of plant tissues with external water. Okay, it's not going to, if it is on top of the lamina, the leaf lamina, it's not going to diffuse through the leaves because of this waterproof cuticle. So that's how it serves to prevent contamination. Okay, contamination with the external water, dirt, or even pathogens or microorganisms that we have mentioned just now. Okay, so another importance is that glycolipid. Okay, so this is the structure of the plasma membrane. Again, you have seen this back in your chapter 3. So you get to see the phospholipid bilayer, these embedded structure here, those are the proteins, and that you get to see this antenna coming out like that. So if this emerges out from the head of the phospholipid, so from the head, you call it as the glycolipid. Glycol here refers to the carbohydrate component. Glycolipid. Lipid here refers to this as a lipid structure. Okay, the phospholipid. So the main function is to ensure stability of the plasma membrane, also to help in cell identification process. Okay, so they may act as a receptor. Okay, they might be responsible for specific cellular contact, okay, or signal transduction. Okay, that's the function. Example of functions stated for glycolipid. Okay, next is on cholesterol. It is important in steroid hormone synthesis. So on the basis of receptors, their receptors, steroid hormones, they have been classified into 5 groups, namely, okay, so you can see over here the listed 5 groups of the steroid hormones. Not to worry about this, not to be burdened by all these chemical names over here. Just look at the top one here. Okay, which concludes this, the cholesterol, you see, it serves as the important precursor or starting point for the synthesis of these steroid hormones. Okay, so it starts with the cholesterol. Okay, so you have come across a few importance of lipids in cells. Okay, reserves, that's the one. Okay, so this is the summarized of all together what we've discussed just now: reserve energy, okay, protect internal organs, cuticles, sebum. Okay, sebum just now. Yeah, here it says sebum secreted by our skin also consists of wax. Okay, so among the importance of lipids, here are the listed ones. Okay,